Hi, this is Science Hangout from Science Mission. From Science Mission. So today we have Dr. Chris Deppmann, Associate Professor of Biology from University of Virginia with us. Dr. Deppmann has published several outstanding work in very prominent journals. Also, he's in the editorial board of several journals and has been rewarded several grants for his. In 2016, he won All University Teaching Award from University of Virginia. In 2015, he was named Cavalier Frontiers Fellow. 13 and 14, he was made honored faculty. He won a Hartwell Foundation Foundation Individual Medical Research Award. 2011, he won the Fund for Excellence in Science and Technology Junior Faculty Grant from University of Virginia. So these are some of the few awards he won. Dr. Deppmann, welcome Dr. Deppmann to Science Hangout.
Oh, thanks very much. I appreciate the invitation.
Let's talk about your background, your education. Can you talk something about little bit about your background and education and all?
Sure. So I grew up in Michigan. I was born and raised in Ann Arbor, kind of in the shadow of University of Michigan. I didn't end up going to University of Michigan. I ended up going to Western Michigan University. But throughout high school, I kind of sought out a bunch of different experiences, including working for an ambulance company. And what I found was I didn't actually enjoy dealing with the patients, but I really liked the science behind what we were doing. And so that drew me to study science at Western Michigan University. And I was fortunate to get an undergraduate research position and studied, signal transduction, how cells talk to each other in the context of toxicology. And so at that point, I just really liked signaling. And so when I graduated from Western Michigan University, I went to Purdue University, which had a really strong cancer center and a bunch of people working on really interesting signal transduction mechanisms, cell-cell communication. And so there I was working on how basic Lucene zipper transcription factors could influence viral progression, Epstein-Barr virus, immune cell development, and cancer formation in the context of signal transduction. I finished up that work, graduated with my Ph.D. in biochemistry and molecular biology, and I was like, well, I still really love signal transduction, so but I want to learn something different. And so, what is the kind of emerging field? What's really exciting? And so then I went to Johns Hopkins to study with David Ginty, and he works on, we now work on the development of the nervous system. But really what caught me was the fact that these neurons are really, really long. And you can actually start a signal down here and watch what happens down at the cell body. So this idea that you can separate signal transduction means it's like the perfect system to study everything that I'm really interested in. And so that's kind of how I got there. So from this little high school in the shadow of Ann Arbor to Western Michigan University, which is a smaller college, to Purdue, to Johns Hopkins, and now here at University of Virginia.
Okay. So do you have any role models or like mentors who helped you throughout your career?
Yeah, I really think that all of the mentors that I've had have been amazing, and it's really humbling to think about, their generosity and everything that they've done for me, starting with my undergraduate research advisor, Leonard Ginsberg, my graduate mentor, Elizabeth Taporowski, to my postdoc mentor, David Ginty. They've all been incredibly, incredibly generous with me. And, I feel like, I'm really kind of standing on the shoulders of giants, not only in in terms of building on the science that we all developed together, but also kind of passing on that spirit of generosity to the people that I mentor. So I think actually that's a really important question. Mentors are very, very important.
Okay, I know you are a busy person. Being an associate professor, you need to write grants, get money, take care of the projects and all. So just I want to figure out, what are your hobbies and interests?
Well, so I really love to cook. I think that cooking is not so different than biochemistry. You got to kind of put the different ingredients in. You got to balance the acid and the basic and the salt and to make something that tastes amazing. I think there's also a really interesting neuroscience component when you're tasting something delicious, and it's evoking like this memory of your childhood or something like that. It just creates this kind of powerful experience. So I think everything about cooking is fascinating to me. I don't do this very often anymore, but I used to really like to play poker. Cards, I think that kind of psychological aspect and the math coupled with the math aspect is really interesting. I love books. I love movies. Like hanging at home with the dogs. Hiking is fun.
Okay. And let's talk about your lab and lab people. So can you give us a little bit overview of your lab and the grad student postdocs? How do you interact with them and all?
Yeah, so right now we have between 6 and 8 grad students. And the reason it's fuzzy is because there's a couple co-mentored graduate students. And that's something I'm really enjoying more and more, where one grad student is both in my lab and somebody else's lab. So we have one student who is in my lab and somebody else's lab who studies endosomes. And then we've got another graduate student who's in my lab and somebody in biomedical engineering who's finding these really interesting ways to kind of measure multiple things at once by like flow cytometry. So we can actually measure 50 markers simultaneously, and we think that this is going to really help us to rapidly assess what's going on in different mouse models and things like that. So between 6 and 8 grad students, we've got a couple postdocs, a lab manager who saves my bacon on a daily basis. She's keeps the lab running. She's awesome. A senior lab researcher who kind of helps everybody along with biochemistry experiments and things like that. And then, I think part of our mission, this is an undergraduate university, is to teach undergrads. So we always take undergrads in their first year, and they're usually with us all 4 years. And so we've got about 50 undergrads in the lab now, too. So the lab is actually around 28 years.
Wow. Let's talk about your research, and can you present your work, like whatever you are working on, the neuroscience, neural development and all?
Yeah, sure. I think, and I think we're going to get into this in a little bit. I can give you some slides, but basically the idea behind my lab is that I am really interested in how the nervous system is assembled during development. And in particular, I'm interested in the rules underlying that assembly of the nervous system. We've got billions of neurons and trillions of synapses, but only 23,000. And so understanding, so every connection can't be barcoded. There actually has to be kind of a general organizing principle. And the organizing principle that we talk about a lot, we think about a lot, is competition. So competition everything is overproduced, and some neurons persist and survive, and other neurons die if they're not connected properly. This happens for axons and synapses and things like that. So by understanding that basic principle underlying nervous system development, we're interested in all of that. We think we're starting to apply that to diseases. So things like metabolism, pain disorders, axon degeneration, both in the context of like spinal cord injuries, but also Alzheimer's disease and Parkinson's, like that. So yeah, we take kind of a broad view of everything, but it's fun.
Okay, let's see your presentation, if you don't mind.
Okay. So let's see if I can figure this out. Do the screen share, and I'm going to go ahead and do application window. Sure. Okay, good.
Yeah. Good.
So perfect. Okay. Let me see if I can find my keyboard here. Good. So I'm going to go ahead and just play this.
Yeah.
Okay. Okay. So essentially, as I mentioned, we're interested in the rules governing the assembly and the disassembly of the nervous system. Basically, our big question is, how is a nervous system assembled? And we've got this problem that we have to achieve complexity with limited genetic information. And so, as such, we have to reduce, reuse, and recycle the genes that we use. And so the rule that we think about a lot is competition, as I mentioned. So this idea is actually kind of old, and this is one of the only biological papers done by Alan Turing, and he was kind of the was really critical for winning World War II and the father of artificial intelligence. But he did write one biological paper, kind of mathematically modeling how two opposing morphogens can cause pattern formation, complex pattern formation of almost anything. And this is particularly relevant to biological systems. The only catch here is that these two morphogens had to have very different diffusion coefficients. So later on, these two scientists, Meinhardt and Gear, came along and they said, well, actually, we can build in some different feedback loops. We can get even a greater diversity of patterns formed. But then we don't need these, we don't need very different diffusion coefficients. We can have diffusion properties very similar to each other. And so that's kind of where we started. And so when you think about it, if you look at around nature, you see these common patterns in nature. Lightning strikes, the arbor of a tree, dried riverbeds, a Purkinje neuron in the cerebellum. And this is all this can all be explained by this thing that first Turing and then Meinhardt and Gear put forward, where we can have kind of the self-activation and lateral inhibition of two kind of opposing morphogens. Now, in this case, obviously, if you're talking about a biological system, we're going to have opposing signaling pathways or something like that. But in the case of trees or sand dunes or riverbeds or things like that, the forces are going to be different. They're going to be things like wind and water drying and things like that. So we're applying this idea to several questions. One is thinking about competition underlying nervous system assembly. So we think about this in the context of survival. Everything is overproduced, neurons, axons, synapses, and then refined back. In effect, they compete with each other. We think about this in terms of axon growth and guidance, axon patterning in the target, so down there is a heart, synapse formation, branch elimination, and all this kind of comes together to create a functional circuit, and then we call this systems matching. On the other hand, we think about how these pathways can be hijacked, kind of like in cancer, to give us normal function or disease, like injury-induced degeneration, which is going to be what I'm going to talk about today, sensory perception, or even fat metabolism. Okay, so if we think about this activation-inhibition paradigm in the context of a population, it's easy to think about how we can get pattern formation. We've got some winners and some losers. But to really understand this, we actually have to look at it at the level of the individual. So at the level of the individual, this could be a synapse, a cell, or an axon. We've got a balance where sometimes the activation wins over the inhibition, these two opposing morphogens. And when the activation wins over the inhibition, we'll get survival, synapse formation, axon growth, or increase in metabolism. Sometimes you'll have a balance between the two, which is going to dictate the excitability of a neuron. And sometimes inhibition is going to win, where you're going to get cell death, synapse restriction, axon degeneration, etc. So let's dig into activation first, where activation wins. and kind of really start to assign, which particular pathways are associated with these feedback loops. Because right now we're dealing with this totally in the abstract. So this is really kind of a classic experiment done by one of my scientific heroes.
I can't see the slide moving.
Oh, you can't see the slide moving?
Yeah.
Okay, let's see. Let's see.
Yeah, no,
it's good now?
Yeah.
Okay, can you see it?
Yeah, I can see the slide. The next slide.
Okay,
Yeah.
good. Okay. So can you see this one?
Yeah.
Okay, good. So this is my hero, Rita Levi-Montalcini and Victor Hamburger, two heroes. They did this really critical experiment, which really informed how we think about nervous system development. They took a chick growing in an egg, a chicken growing in an egg, and you can actually cut out a little window of that egg, and you can actually take off a little piece of the embryo where the limb would come out. If you then suction through the spinal cord, you can kind of see, okay, well, early on, there's lots of motor neurons. And then later on, if I, on the side where I cut off the limb, now there's, 90% of the motor neurons are missing. On the side with the limb intact, only 50% are missing. On the other hand, they did another experiment where they added an extra limb bud. And they see that on that side, there's only 25% of the neurons missing. But on the control side, there's 50% missing. So this told us several things. Number one, neurons are overproduced. And this is going to be a theme. Everything is overproduced in the nervous system early on. A target-derived factor exists. And implicit in this idea is the notion of competition. So the factor that they ended up finding that is responsible for these types of behaviors is a factor called nerve growth factor. And Rita Levi-Montalcini and Stanley Cohen won the Nobel Prize for this. And here they're taking an explant of hunks of neurons, and they see that in the presence of NGF, you get this beautiful halo of axons, and a lot of the neurons survive. But in the absence, there's nothing. NGF binds to a receptor tyrosine kinase. It's a member of a family of growth factors. BDNF, we hear a lot about in the context of depression and anxiety and things like that. NT3, NT4 are also structurally related, and they all bind to these specific receptor tyrosine kinases. Now, this is kind of the field that I get really excited about and thinking about signal transduction and how cells communicate with each other, but I don't want to blow the audience away with intracellular signaling. But instead, I just want to distill this down to this basic question. And this is actually what got me into neuroscience in the first place. How do we actually have NGF coming specifically from a target transmit a signal all the way to the cell body? How's the NGF TrkA signal transduced? And it turns out that NGF is coming from targets like the heart or the eye or the salivary gland or fat. And it can then internalize this entire complex into this endosome. Okay, so think about like packing your entire family up into a station wagon. You're bringing all your favorite stuff with you, and then you get on the road. Your car is the diamine, dinactin. You get on the road, which is the microtubules, and you take it all the way to California with all of your stuff. And so that's all these things like P85 and PLC gamma and RAF and that kinase. You get close to the nucleus and you activate transcription. And actually, here's another way to look at it. So NGF coming from the target. This is a nice figure done by Bill Snyder and Jason Newbern. And basically,
Chris, I don't see that slide. Is it?
How about now?
Oh, okay.
Okay. So maybe
now I can.
I sometimes have to click off the full screen mode.
Okay.
Good.
Okay.
Okay. Okay. So in this case, instead of activating transcription to promote survival, the NGF TrkA signaling endosome can go into the dendrite to promote synapse formation. So clustering of the postsynaptic density, all the neurotransmitter receptors, and then that's sufficient to drive presynaptic specialization. This is based on a paper that we published a few years ago. Okay, so we've also found proteins that are associated with the the signaling endosome. Coronin 1 is one of them, and it's really important for this kind of survival effect and this competition for survival. Okay, so that's the activation. We've got TrkA and coronin on one side. What about the inhibitory side? So for this, it seems that in terms of this P75, it's a member of the tumor necrosis factor receptor class of family. And basically anything that TrkA is doing, or TrkB, P75 is usually doing the opposite, at least in the sympathetic nervous system. And so we're thinking a lot about these interactions. So on one side, NGF-TrkA, and on the other side, P75. In sensory neurons, this relationship changes. It's P75-TrkA on one side and TNF receptor on the other, which is a close cousin of P75. In non-peptidergic sensory neurons, it's RET on one side, and we're not exactly sure what's on the inhibitory side. And then in proprioceptive neurons, the neurons that kind of sense your body's position in time and space, we have TrkC on one side and TNF on the other. We also see this phenomenon called reverse signaling, where the ligand actually signals back into the cell. I won't belabor this too much. Okay, so that's kind of the groundwork for how we think about the assembly of the nervous system. What I next want to talk about is the disassembly of the nervous system and how we think about degeneration. Okay, so degeneration isn't always bad. In fact, most of the time it's good. If we don't have degeneration, then we can't form a functional nervous system. Everything is overproduced. We produce way more axons than we need, and we actually have to sculpt these back to make a functional nervous system. And so that's what we see in the developmental branch elimination. But sometimes the more popular, the more, not popular, but the more well-known aspects of degeneration are from either injury, like if you get in a spinal cord accident, or from neurodegenerative disease, like Alzheimer's, Parkinson's, Huntington's. Okay, so let's talk about
slides.
Huh?
Slide. I just see the NGF slide.
Okay.
Yeah.
All right.
Okay.
Here we go. Okay, so let's talk about the developmental side first. So during development, axons can sometimes send out multiple branches to different targets. And it turns out that if you have a little bit better connection over to one target than the other, so like here I'm showing more NGF than the other side, we'll get the side with less NGF disintegrating. And what we know is that antagonistic receptor, P75, is required for this. So if we knock out P75, we don't get this at all. And so that actually fits nicely with our kind of activation versus inhibition paradigm. And that's what we see here. We also know that P75 is part of a family, at least in this sympathetic nervous system, which controls our fight or flight response. It's one of three that are primarily expressed. TNF receptor 1 is the other one. Death receptor 6 is the other. And we see that they're all clustered in the same clade in this phylogenetic tree out of 27 TNFR family members. So highly similar, all expressed in the sympathetic nervous system. We see that, so we wanted to look at these other factors, and so we looked at sensory neurons, and so what you can see is that in the absence of TNFR, these are TNFR knockout animals, or the ligand for TNFR is TNF, tumor necrosis factor. We see that we get tons of axons going into the paw, and these mice actually experience quite a bit of pain because they're producing way too many axons. Also, Marcus Levine and colleagues have shown that DR6 is required for developmental degeneration as well, and that's in the bottom panel.
Because this.
Yeah. Can you see it now?
Same slide, right? Okay, now I see. Okay.
Okay, good. Okay, so in the context of development, we've got TrkA versus all of these TNFR family members. Okay. What about in the context of something pathological like acute injury? Do the same rules apply? Well, it turns out that there's no receptors that have been described for injury-induced degeneration, so this would be really strange. There's actually quite a bit known about injury-induced degeneration, which is also called Wallerian degeneration. We know that there's a metabolite called NAD that is rapidly depleted, and that seems to be quite important for degeneration, because if we actually overproduce this compound NAD, we can preserve these axons for quite a bit of time. It seems like this protein SARM is responsible for chopping up NAD. And so we know that if we knock out SARM, we can preserve axons after injury. And this seems to be going through some other pathways that ultimately lead to degeneration. And there's all these kind of interesting feedback loops going on here. But SARM was actually first identified as something that's associated with the receptor, the toll-like receptors. And so in the context of injury, there's never been a receptor shown to be associated with SARM. They think that this is totally happening within the axon. They just kind of self-destruct, okay? But if we go back to our developmental model, we know that there are receptors involved there. So where's the disconnect here? You know, are there actually similarities between these two types of degeneration? And so that's a question. Can we get the similar balance between receptors, these cell surface receptors, in the context of injury, where that's never been described? And I just want to take a second to acknowledge all the people that have done this work. This recently came out in Current Biology a few weeks ago. I think that this is going to be an area of research for us in the future. Okay, so let me just tell you a little bit about the experimental setup. Oops. What happened to my thing? Okay, so the experimental setup. We grow. Can you see it?
No.
Okay. Let's see. Can you see it now?
Yeah. Yeah.
How about now?
Yeah. Now
Good.
I can see.
Okay. So we grow sympathetic neurons in microfluidic devices. Think of these as microchips for neurons. Okay. They grow across these little microgrooves, and so we can, in effect, separate the cell body and the axon. Okay, we can either deprive these guys of nerve growth factor, which is going to elicit kind of a developmental degeneration type thing, which is also very similar to Alzheimer's type degeneration, or we can take an aspirating pipette and a vacuum and just vacuum off the cell bodies and leave the axons intact, and this would be like a motorcycle or motorcycle accident or something like that. So this is what the injury looks like. We can just rip off those cell bodies. And you can kind of see on the right-hand side, the axons remain intact. Okay? After we rip off all the cell bodies, we can throw those neurons, these plates, back into the incubator and just essentially watch how long it takes for these axons to degenerate. And so this is essentially what we'll count. We'll stain for something in the cytoskeleton like microtubule, and we'll basically look for things that are beading and blubbing. And if they've got more than 1 or 2 beads of blebs, we count that as degenerated. So, for example, in this picture, we would consider this 90% degenerated because there's one of these boxes that has 1 bead, which we wouldn't consider degenerated. So that's how we count. We've also figured out how to automate this, and so this is an example of like healthy axons, and we've got software that can automatically scan this, and then we can throw this into a program that can automatically assess degeneration. This isn't quite ready for prime time yet, but so we're still kind of counting by hand. Okay, so this is what it looks like comparing NGF deprivation to injury. If you take away NGF, it takes quite a while for the axons to look all mangled and blobby. So by 24 hours, we get quite a bit of degeneration. In injury, this happens much faster, and in fact, we can quantify that. We've got the NGF deprivation. It takes about 24 hours to reach that 50% degeneration point, but in the context of injury, it starts happening right away. The kinetics don't change as a function of NGF being present or whether we're using juvenile or adult neurons.
Can you move the slide, please?
Sure.
Yeah.
Okay. Good?
Yeah.
Okay. So we wanted to compare the relative contribution of TNFR family members in development versus injury contexts. And what you can see here is that we just did the same experiment using neurons from TNFR, P75, or DR6 knockout mice. It might be hard to see on this, but we can quantify it here. And we basically see that in the absence of TNFR, neurons degenerate just fine. In the absence of P75, they're protected pretty well. They will eventually degenerate, but at the 24-hour time point, they're protected. DR6 protects them a little bit. These mutants are involved in injury-induced degeneration, and they also have some modest protection in this developmental paradigm. Yeah.
Can I see? Can we see the slide, please? Okay. Yeah.
Good. Okay.
Yeah.
So this is what we think is happening in development. DR6 and P75 are working in parallel to promote degeneration. Is that also true in injury? And so, sorry, we can pull it back. So is that also true in injury? We did all the same things, and then we quantified it, and we see that neither TNFR or P75 is required for injury-induced degeneration, but DR6, like WLDS and SARM, are absolutely essential. This is actually, and we did the full time course here, and you can see that DR6 really extends the life of these axons, as well as any known factor involved in degeneration. And we can do this in sensory neurons too. So it's not just restricted to sympathetic neurons, but also works in sensory neurons. DR6 is totally protective. So this actually tells us that in development, DR6 and P75 are both required, but in injury, DR6 is absolutely essential. And I think the fact that we've got now a receptor involved in injury-induced degeneration is kind of profound, and really is going to change the way we think about how injury-induced degeneration occurs. Can you still see the slide? Good. Okay. So we've got here we've got a sciatic nerve injury model. So it's great that we can do this in vitro, but we want to be able to do this in vivo as well. So in this case, we basically did a surgery on animals and clipped the sciatic nerve, took out a chunk of it, and then sewed the animal back up, and then came back 2 weeks later to watch the degeneration. So this is going to be that distal part, the part towards the toes that give us this degeneration. And what you can see on the left is a stain for myelin. So if you see these kind of little donuts, that means that we've got nice myelinated fibers. And then on the right, we've got electron microscopy, uninjured versus injured. What I think you can appreciate right away is that in wild type, the nice, beautiful axons, these big bundles, are nice and intact. But in the injured, 14 days later, they're totally trashed. So the axons are completely gone. In this WLDS mutant, or in the absence of SARM, the axons stay preserved. And then in the absence of DR6, just like what we saw in vitro, we get a pretty good preservation of the axons. What's interesting here, though, is that in injured, if you look at the bottom right corner, we get a pretty massive loss of myelin. Okay, so there seems to be the axons are preserved, but the myelin is lost, which is kind of a new phenotype, and it suggests that the loss of myelin and loss of axons are separable pathways, which is exciting. Let's skip this. So essentially, what we found is that DR6 is required for injury-induced degeneration, but the question is, does this also feed into these known pathways like the junk pathway? So essentially, we did our axotomy again in vivo, and then we just make sure that we're. So we did our axotomy again, and 30 minutes after the injury, we get a nice induction of Jun kinase, but in the DR6 knockout, that's totally gone. So what this indicates is that after injury, we have DR6 feeding into Jun kinase. And so maybe what we are doing is just kind of expanding the known pathway. But what we don't know yet is what the ligand for this receptor is, and we also don't know how it is that the axon is communicating with the Schwann cell glial cell, and whether there's a receptor to receive that. And so with that, I will end, and I can take questions if there's time. Get out of the screen share here. Okay, good. Hey.
Hi.
Hi. So presentation is done.
Okay, great. So it was a nice talk. Questions like there are those you said like axon degenerates on its own, right? Like
yeah.
So there is like is there any death receptor or anything like that?
Yeah. So that was kind of the main finding is that instead of this kind of old idea that the axon degenerates on its own, there is this death receptor 6, that is absolutely essential for the degeneration.
Okay. And do you think that there would be other receptors in addition to the death receptor, like what you found out in different tissues?
I think that would be really interesting. I mean, thus far, based on our in vivo model, we know DR6 is essential for the degeneration of sensory neurons, sympathetic neurons, and motor neurons. What we're thinking about now is whether it's also going to be essential for degeneration in the central nervous system, and so we're doing those experiments right now. Whether or not there's other receptors involved is a really interesting issue. The point that I think I failed to make is that by now knowing that there's a receptor and probably a ligand for this, it might give us an explanation for why it is that we get bystander degeneration. If you have a stroke or a traumatic brain injury, if you have a little region of the brain that is actually injured, why is it that there's this whole halo around the injury site where neurons are lost also? So this might provide an explanation for why you get these bystander neurons or bystander axons being degenerated. And so in that context, I think it is possible that there could be other receptors involved, maybe toll receptors or something like that.
I don't think I have any other questions, but it is a very interesting, intriguing presentation, and I read your paper too. It is pretty, it is really something new you are finding out there. And thank you for making this effort to present your work to our audience, and wish you good luck, and hopefully we will be able to catch you sometime soon again with some new publication.
Awesome. Yeah, it was a pleasure talking to you. I appreciate the invitation again.
Thank you.
Okay, bye.
Bye.